Harmful algal blooms can threaten human health if the organisms present produce toxins. We will look at a procedure to detect some of the toxins produced by blue-green algae, or cyanobacteria. Toxins released by cyanobacteria have been implicated in cases affecting human and animal health. Diagnostic procedures have been developed to detect the presence of specific types of toxins. We're going to demonstrate how to measure a particular group of toxins following the Ohio EPA Total Microcystin Analytical Methodology using an ADDA ELISA kit. ELISA stands for Enzyme-Linked Immunsorbent Assay. The acronym ADDA refers to a part of the microcystin molecule that, when measured, serves as an indicator for the majority of the various microcystin variants, or congeners. The assay involves collecting a water sample, rupturing or lysing any cyanobacteria cells, and detecting the specific toxins using the sensitive antibody-based ELISA technique. ELISA produces different color intensities depending on the toxin concentration. Please refer to the Ohio EPA method to learn how to collect, preserve, and prepare samples for ELISA analysis. These important steps are not included in this training. We are starting with individual samples that have had any cyanobacteria cells ruptured or lysed. Please note that the ELISA described here is specific for detecting a broad range of microcystins containing the ADDA structure and a related toxin, nodularin. Your ELISA kit may be slightly different and may be tailored to a different group of cyanotoxins, but the equipment, consumables, and procedures will generally be similar to those described here. Organize the ELISA equipment, including a 10 to 100 microliter adjustable pipette and a 300 microliter multi-channel pipette. Bring the plate, reagents, and standards to room temperature, 20 to 25 degrees Celsius. Dilute the wash solution concentrate to the manufacturer's instructions. Please refer to the Ohio EPA method to learn how to perform the quality control and calibration steps. Determine the number and type of quality control samples before following the manufacturer's schema for plate setup. The accuracy of ELISA depends on the analyst technique, including pipetting sequence, accurate reagent volumes, and consistent incubation periods. Being careful not to touch the bottom of the well, load the ELISA plate with duplicate 50 microliters of standards, quality controls, and samples into their predetermined well locations. Add 50 microliters of the antibody solution to each well with a multi-channel pipette. Cover the wells with adhesive film and mix the contents with a rapid circular motion for 30 seconds. Be careful not to spill the contents. Start the timer for 90 minutes. Incubate the samples at room temperature. After the 90-minute incubation, remove the covering, turn the plate upside down over a sink, and vigorously shake the contents out of the wells. Using a multi-channel pipette filled with diluted wash solution, generously rinse each well. Remove the wash solution from the wells by aggressively striking the inverted plate on absorbent towels until there are no bubbles, droplets, or half moons in any well. Hold the plate up to the light to see if any solution remains. Repeat the rinse process a total of 3 times. Add 100 microliters of enzyme conjugate solution to each well using the multi-channel pipette. Cover the wells with the adhesive film and mix the contents with a rapid circular motion on the benchtop for 30 seconds. Start the timer for 30 minutes. After the 30-minute incubation at room temperature, remove the cover and vigorously shake the contents of the well into the sink. Wash the wells as before by filling the wells with diluted wash solution and striking the inverted plate on absorbent towels. Hold the plate up to the light to see if any droplets or bubbles of solution remain. Repeat the rinse process a total of 3 times. Add 100 microliters of substrate solution, sometimes referred to as the color solution, to each well using a multi-channel pipette. Fill the remaining wells systematically, in an order and at a rate that can be repeated in the next step. Cover the wells with adhesive film and mix the contents with a rapid circular motion for 30 seconds. Be careful not to spill the contents. Protect the plate from light. Start the timer for 25 minutes. The antibodies specific to the toxins link together with the enzymes and create a color change. The darker the color, the lower the concentration of toxins, and the lighter the color, the higher the concentration of toxins. Please note, the color intensity is inversely proportional to the concentration of toxins. After 25 minutes, add 50 microliters of stop solution to each well using a multi-channel pipette. Before 15 minutes has elapsed, measure the color by reading the absorbance at 450 nanometers using a microplate ELISA reader. Most of the ELISA plate readers will have software available to calculate the values for the calibration curve and determine the toxin concentration in the sample. The analytical equipment, software, and hardware can be assembled as a mobile unit. Such a kit can be rapidly deployed to sites where cyanobacteria blooms are occurring and ELISA testing is needed. The analyst operating a mobile unit will follow the same procedures that were demonstrated in this training. To determine each analyst's capability to meet the appropriate qualifiers, QC acceptance, and method reporting limits, please refer to the Ohio EPA method.